May I introduce our next speaker, Dr. Aragon-Martin? Is Jose here? He's our long-standing molecular geneticist, and he has overseen the mapping of the Marfan gene in the United Kingdom for over 300 families. He's also directed our Sonalee Laboratory program of research to find new genes and conditions which overlap with Marfan syndrome and can sometimes be confused with them and make diagnosis difficult. So to clarify this, over the years, we've looked at several other conditions, and Jose is going to tell us about his approach. Is he here?
Jose's here.
Yes.
Yeah.
Let me just share my screen now.
Hello.
Hello.
Hola.
Hola. There you are. Can you see my screen? Yes? Good.
We can see a screen.
Let me do it like this. It will be easier for me. Okay, thank you for your kind introduction. First, I would like to acknowledge that we have moved from St. George's, University of London, to Imperial College in October 2019. The name of the lab was taken from a graduated medical student who had Marfan syndrome. But actually, everything started from the Marfan clinic. Patients will come to the clinic with various conditions involving the cardiovascular, like thoracic aortic aneurysm, ocular problems like ectopia lentis, or skeletal problems like scoliosis. And that is how we got interested in other related conditions. But first, we learned more about Marfan syndrome, and we started to screen patients who came to the Marfan clinic. That was back in 2000. Those who were negative for the FBN1 gene were left without an answer. But through the years, we kept these families with us, and when we had funding, we started to research on other genetic biomarkers on the related phenotypes. We started to work on thoracic aortic aneurysms and dissection, also called TAD, ectopia lentis or EL, scoliosis, and Ehlers-Danlos syndrome or EDS. In Marfan syndrome, we have been trying to look for the best tests to be used for the screening of FBN1 gene. First, we started with SCCP back in 2000, then DHPLC around 2005, Sanger in 2012, and now 2020, we are doing it with NGS. We are looking at the possibility of associating a particular mutation with a clinical feature, studying genotype-phenotype associations in Marfan syndrome. This will also help for the fine mapping of the FBN1 gene, where we want to establish which variants are causing Marfan syndrome and which variants are just that, normal variations of the same protein. We are also collaborating with a group in Cambridge for a new Marfan syndrome drug discovery. And as a future project, we would like to study by the gene editing, the functionality and expression of unknown significant variants. For the TAD project, we have about 21 large families and 33 small families. We have been using NGS to discover new genes in these first 3 TAD families. This is the first family here on this picture. This is the pedigree of the first TAD family with the green circles, the samples that we use for the analysis. 2 over here on the left and 1 over here on the right. First family had 5 candidate genes. The second and the third families are under NGS pipeline process at the moment. Our research on the first TAD family has discovered a new gene called LMOD1, and it will be published in 2020. A zebrafish model has been studied when blocking the LMOD1 has produced a phenotype that might influence in the formation of the thoracic aortic aneurysm seen in TAD patients. First embryonic pictures shows a curvature of the spine and also other phenotypes like oedema and reduction of the eyeball. If you can see on this group of pictures here, the first column here is from the normal fish, also wild type. In the middle here is the moderate affected, and over here is the severely affected on the right-hand side. And the first row is for the spine. The second row is for the oedema, which is just accumulation of the fluid. And you can see that from this red arrow here and here for the severe. and also the ocular here on the bottom, which you can see that this has reduced. This is the normal eye, and it has been reduced very much on the severe form. Also, we look deeper into the formation of the aorta. There are regions in the zebrafish model with Elmore-1 block that has not developed yet to the normal extension. And you can see that on this bottom left-hand side pictures. On the left one here is the control or the wild type normal. And here on the right-hand side is the block gene. There's some extensions here on the, when we can see in the control, one here and one here, for example, that it doesn't appear on the block Elmore-1, shown by the arrows here. We are working on future studies in Elmore-1, where we want to study other variants using the gene editing technique to increase its responsibility with TAD. Rescuing the wild type, known as the normal type, will help us to understand how this gene works and also how the rescuing technique works, so we can use it in other related conditions. We want to compare phenotypes using zebrafish as the animal model with other TAD genes, so we can build a more robust understanding of how TAD is formed. Sonalee Lab has about 51 TAD probands that needs to be NGS screened, and at present we are evaluating 2 more families with 2 candidate genes, both linked to the degradation of the extracellular matrix or affecting the structural integrity of the ascending aorta. One of the questions that I get from patients is, what do you study other genes that has nothing to do with Marfan syndrome? My response is that we cannot say that none of the other genes are involved with Marfan syndrome, but we can say that there are genes that interact with FBN1 gene in the TG-beta pathway. And studying this genetic aspect of these genes might give us a better understanding of the pathogenesis of Marfan syndrome and perhaps the different clinical features appearing in Marfan syndrome. As you can see from the TG-beta pathway here, number 1 is fibrillin-1 in blue here. And there's another gene here called, it's a protein, LTBP1, and is bound to the fibrillin-1. And the whole complex here is called LLC, which sequesters the inactive TGF-beta. When it's released, it becomes active and is bound to the TGBR2 receptor, which activates the TGBR1, and it produces a cascade of in this pathway. And if there is a mutation on any of these genes, it will form this aneurysm or dissection at the end. The same thing that has been seen in Marfan syndrome. Also, there are genes that belong to a different genetic pathway, but share similar clinical features seen in Marfan syndrome, like thoracic aortic aneurysm and dissection, the TAD form, or even ectopelentis, also known as EL. Studying these other pathways on similar clinical features might bring a better perception of how they appear in the first place. This is another way of formation of thoracic aortic aneurysms is the smooth muscle cell contraction pathway. And in the picture here, we can see number 1, which is the fibrillin 1, and picture number 2 here is the collagen 3A1 protein. ACTA2 is seen here, 4 of them, and here as well. And number 4 here is the NYH11, it's another protein for TAD. And on the right-hand side, we can see the ACTA2, also the NYH11, but the NYLK gene, protein in this case, is number 5, surrounded this one. And again, mutations in these genes develop thoracic aortic aneurysm, or also seen as in Marfan syndrome. It is imperative not to miss a thoracic aortic aneurysm causing mutation. If the patient comes for genetic screening, we should be prepared to give the best we can do to find out the genetic cause, since not every gene has the same predicted progress in clinical features. It will help to rule out Marfan syndrome if we can link a patient with a particular gene mutation. That is why we are doing research in ectopelentis. For instance, we have this EL family with a negative FBN1 gene mutation and with a possible positive ALN-TSL4 mutation. Bear in mind that mutations in FBN1 gene are dominant, while mutations in ALN-TSL4 are recessive. You can see this family here, which is a consanguineous family. Patients with this black dot means that they are recessive. Sorry, it means that they are carriers. And this patient here is normal. Those with the black shaded picture, they are affected, which is one very interesting family actually, because everything fits very well except with this patient here. It carries the mutation, but it doesn't express it. So we don't know if either being misdiagnosed. That's obviously from a point of view of researcher. Misdiagnosed at the point of the ophthalmologist, maybe is an incomplete penetrance, which basically means that you carry the mutation, but you will not express it, but you are able to pass it to the next generation. Or it could be that actually this is not the gene, but we're gonna do some research on this one. Our continued collaboration and interaction with other groups around the world has enlightened us to understand now that ADAN-TSL4 and FBN1 protein forms part of the zonule fibers seen here in both of these pictures. On the right-hand side here, you can see the zonules here in this electron microscopy picture. This is the lens over here, and these are the zonules. This is a picture of a normal tension of the zonules. Here in the center, picture also shows zonules being detached from the normal section in the eye, and the lens has been misplaced. These are the fibers here. As you can see, there's some sections has been missed there. Our collection of FBN1 negative EL patients has moved to the project forward when also ADAN-TSL4 mutation screen was negative. We have a collection of 28 families with ectopia lentis with no gene associated with them. We know that there are several genes involved in EL, but just screening by Sanger is very expensive. So our research approach is to find new genes using NGS analysis. If a new gene is discovered, then we will use an animal model to mimic the phenotype and understand better how EL appears in the first place. Also, we want to research if we can revert the mutated form into the normal form by gene editing. And later, this can be translated to the benefit of the patients. Just I want to mention briefly that we are also researching on scoliosis. We have several families, and we're studying at present 2 of them. We have found a candidate gene in the first family with the help of an animal model, and the paper should be published in 2020. These 3 videos will show the rotation of the zebrafish skeletal body. The first one, let me see if it works. The first one is from a normal zebrafish. The second is from a mild form of the new gene in zebrafish. And the third is from a severe form of the new gene in zebrafish. You can see from the first video and the second that there is not much difference between both of the animal models, but there is a slightly change on the second. When looking also at the third animal model, then we can appreciate the curvature of the spine. I do not want to forget that we are also researching on Ehlers-Danlos syndrome, in particular the hypermobile version of EDS called HEDS. There's no gene associated with HEDS yet. We have several families, and we are working at present on 2 of them. We have 1 strong candidate gene in the first family, which is under study at the moment. And the known EDS genes in this family needs to be ruled out to confirm the validity of the NGS analysis. The second family is still under NGS pipeline process with 23 candidate variants in 10 genes. Before I finish, I would like to talk about another question I have been asked. Is there any gene therapy for Marfan syndrome? My answer is that we are just starting to grasp what we can do with the gene editing tools that we have right now. There are 2 very interesting papers on gene editing in Marfan syndrome. The first one in 2018 and the second in 2019. Both of them claiming that they are very precise and specific on the base substitution capability. The overall scientific community agrees that there are still problems with the technique used and needs improvement. But overall, I would like to say that the future looks promising. This is my last point. Some of you might be thinking about, is my donation enough? Or where is my donation being used? Any donation is crucial to continue with the research in Sonalee Lab. Time is important, so 1 penny today will not buy you a chocolate bar, but if everybody donates 1 penny, we can start to contribute to buy that chocolate bar. And together we can benefit from sharing that chocolate bar. At the moment, we need funding for TAR and EL projects to discover new genes. When we use NGS analysis, we need at least 3 patients from the same family to progress. At the moment, the cost to NGS, in this case, whole genome sequencing, for 1 sample is £840, but the cost has been dropping yearly. Studying other genes will update the genetic map with other genes that interact with FEN1 gene. The study of these genes involved in the same phenotypes seen in Marfan syndrome might help us to understand better how Marfan syndrome phenotype develops. It will also rule out Marfan syndrome. One of the problems studying gene editing in Marfan syndrome is that the full sequence of the gene in the zebrafish is not well known. Another problem is that target sequences are not very specific to a particular section of the FEN1 gene, and they are shared along the gene sequence and also with other genes like FBN2. So to make it more specific to our target is something that we need to improve. Studying gene editing in other tag genes in zebrafish that has more targeted specific sequences might help to understand how the aneurysm occurs in the first place, and this might help to understand how this occurs in Marfan syndrome. I would like to say thank you to all patients and their family involved in Sonalee laboratory research. Also, let's not forget the volunteers and fundraisers that always give their free time for this cause. Marfan Trust team always trying to help the Sonalee laboratory with their best. Students from all years, medical, PhD, MSc, and BScs, who come to our laboratory to be trained, gain experience and knowledge about Marfan syndrome. And finally, the sponsors, which sometimes give that extra help to push our research a little bit further. And just want to say thank you.
Thank you very, very much, Jose. And I think we all understand what's going on in the lab now a bit better. Can I just ask a very practical question?
Sure.
We have many patients who go through the gene screen at places like the Brompton Hospital. They come in with an aneurysm, and while they're there, we take a blood sample and we see if we can find the gene. A large proportion of these people do not have any one of the 51 genes that we know about now. What proportion do you think are not explained, and how many more genes are there to find?
Based on our, actually I got a slide here. Let me see if you can see that slide. Based on our research, we have about 78% unknown links to the gene of patients. So we're speculating here between 50 to 78 new genes will be there to explain that in our data. So
we still have a lot of work to do.
Yes.
Over to you.
Well, the thing is that we now have two pathways. We have seen the TG-beta pathway and the smooth muscle pathway. There must be something else there as well, or all the genes that belongs to the same pathway, but we haven't found the gene yet. But it had to be one of those as well.
And each of those families is like a Marfan family, aren't they? So
yes.
Since they present in a very confusing way, we have to not only rule them out as not having Marfan, but we need to be working to find out what the cause is, and also better medication. And anything we learn from the other families may actually apply to the Marfan families as well.
Yes, yes, because they belong to the same pathway. So if we can find a way to block somehow and help one of the genes to block the thoracic aortic aneurysm, they should be more or less the same thing happening in the same pathway. So yeah.
Very good. Victoria, do you have any collected questions?
I have one so far. It's arrived in the chat function. I think it's a benefit or otherwise of receiving a definitive diagnosis. Mr. Bruce was offered genetic testing for Marfan syndrome. He has severe eye skeletal indications, but advised by a travel health insurer that if he were to be formally diagnosed, they would not cover him. And until then, he's covered as normal. So he declined the genetic test. After discussion with eye consultant, cannot see any advantage, only disadvantage relating to insurance. He has an annual echocardiogram to monitor the aorta, but neither himself nor his late mother exhibited abnormalities in that area. Is there any interaction with insurance situation to make it unwise? Is to mitigate the blankets exclusions due to the possibility of heart issues?
That is definitely something that somebody else is going to have to probably help me to answer that question.
Yes, I'll start. Anybody else can join in if they want. This is a vexed problem. And in actual fact, in the Johns Hopkins Clinic, if a couple ring up for an appointment to ask if their child has Marfan syndrome, they're told to go away and get life insurance and then come back and they'll be given an appointment. Because once you have life insurance, especially if you can wait a year without claiming life or health insurance, then they pretty much have to look after you the rest of your life. But what's the advantage of actually having a genetic diagnosis? Well, if the doctors are going to make a clinical diagnosis anyway, because you have eye and heart trouble or heart and skeletal trouble, if you have 2 out of 3 systems affected in a classical way, they're going to make the diagnosis anyway. It doesn't matter if you've got a gene discovery or not. So I think probably it's an advantage in England because the NHS covers us from cradle to grave. You can have any treatment you need with Marfan syndrome through the NHS. It's not the same in America, of course, where most health insurance is private. And so the stakes are much higher there. But I think probably having a gene diagnosis is good because you can screen your parents, your brothers and sisters, your children, and make sure that they don't have it as well, perhaps in a milder version that's gone missed. I have several instances in my practice of 3 teenagers who were diagnosed, and when the parents were screened, the father was also affected, and in one case he needed emergency surgery. So I think probably overall in the UK it is advantageous to have a genetic diagnosis. Also, it may be, you may have even an aneurysm, but caused by another gene. And that may be more severe than Marfan syndrome. So you won't get the right treatment or the right medication unless we can classify your aneurysm. Does anybody else have anything to add?
The converse to that is when I worked in Australia, I had a patient who was diagnosed with Marfan syndrome, which affected her insurance. She then had genetic analysis at the age of 45, and she didn't have a fibrillin 1 mutation. And in fact, she just had isolated ectopia lentis, which she'd been misdiagnosed with Marfan syndrome for her whole life. As Anne said, I think the diagnosis is already made, is already on the paperwork. I don't know much about insurance, but I think if the paperwork it says you've got Marfan syndrome, and I don't know how much more the genetic confirmation would make a difference too.
I think in that case, that's the reason we're doing our research, isn't it, Jose? Because
if we can differentiate between Marfan syndrome and mutations, for example, in RNTSL4 for the ectopia lentis, then it will be much better and easier as well. Like you mentioned it as well before, there's this ACTA2 gene, which is severe for the thoracic aortic aneurysm, and it's better to find out first which gene is actually involved in your condition and then be treated and hopefully change the insurance by the fact that if you only have ectopia lentis, this is one of the research we want to do in FPM1 as well. We want to find out with the genotype, phenotype, those patients which have Marfan syndrome. For me, any mutation in FPM1 is Marfan syndrome, but then we have to divide them into those who will only develop ectopia lentis and those who will develop, for example, skeletal, or those who will develop aortic aneurysm. Or it's going to be, like Aman was saying before, even though you have ectopia lentis right now, you have to be checked by for thoracic aortic aneurysm because we don't know yet what it is, but eventually in the future, hopefully we can amalgamate all the information and be able to help.
Can I just say something? Anne, it's Alan Saga.
Oh, hi, Anna.
Speaking from the genetic point of view, where I get asked this question a lot, I think you have to differentiate between it being a diagnostic test, where the clinical diagnosis is established and you've got a working diagnosis, or whether you're doing a predictive test. So if the patient has enough criteria to diagnose it clinically, the gene test is not going to make any difference. If the echo is normal and take another patient, they don't have ectopia lentis, they don't have any other features, and the echo importantly is normal, then that's a predictive test, and that doesn't need to be declared to the insurance in the same way as you don't need to declare a test for breast cancer susceptibility if it's in the family. So I think you need to differentiate diagnostic from predictive. And I would echo your points, and that it's better to know because you can help the other family members, and importantly, there are other genes that can mimic Marfan's, And you do need to make sure you're not missing, for instance, treatment with losartan for low STs.
We have several members who've helped us with research on where to obtain life insurance and travel insurance. And I'm just going to show you briefly a little booklet that they've written. We spoke to Darren McDean earlier, who's still with us, and he engineered this with his friends. So we've done a fair amount of research, and if anybody would like a copy of the booklet together with the information, please do contact us. It's now available.